Good morning, children. Hope all are fine. Let's recall what we studied in our previous botany class. In the previous class, we started with the lesson Classical Genetics. In that, we studied how Gregor John Mendel very carefully and systematically performed his experiments on Phytosativum plant and made numerous observations. He explained the inheritance through monohybrid cross, dihybrid cross, trihybrid cross, and based on the observation in monohybrid and dihybrid cross, he formulated three laws of inheritance. All this we studied in our previous botany classes. In today's class, we are going to study that when Mendel was performing his experiment with the possum plant, other scientists also were working with other plants apart from possum plant, and to their observation, the phenotypic ratio was different from what Mendel has proposed. As we know, the phenotypic ratio for monohybrid cross, Mendel proposed it was 3:1. For dihybrid cross, it is 9:3:3:1 and so on. So here, the other scientists, they also performed the experiments, and their phenotypic observation was different from these ratios. So they explain that this complex pattern of inheritance, they called it as extension of Mendelian genetics. As we have studied genetics, the father of genetics is Mendel. So here they said this inheritance is an extension of Mendelian genetics. So this change in the phenotype, they said this was due to the gene interaction. As we studied, Mendel said each character in a plant or an organism is controlled by one gene. But because of the change in the phenotypic observation, so scientists said that the change in the phenotype was due to the gene interaction. So let's study what is this gene interaction. A single phenotype is controlled by more than one set of genes. As we have studied, Mendel said one character controlled by one gene, but other scientists they said a single phenotype is controlled by more than one set of genes. Each gene has two or more alleles. Already we have studied each gene they has two or more alternate forms, and these alternate forms we call it as alleles. So here they said a single phenotype is controlled by more than one set of genes, and each of which has two or more alleles. This phenomena is called as gene interaction. So here, because of this gene interaction, there are some structural and chemical changes taking place. So many characteristics of the organism, including structural and chemical, which constitute the phenotype, are the result of the interaction between two or more genes. So here they said that is the change in the phenotype is because of the gene interaction. So here the flow chart of gene interaction types are given here. So here gene interaction, so it is said to be there are two types, intralocus and interlocus. Intra, the word intra means within. Locus means the position of gene in the chromosome. So as we have studied, each gene has two or more alleles. Okay. So here it's a intralocus interaction explains that the two or more alleles which is present in the same locus, they interact and bring about the change in the phenotype. Okay. So the alleles which is present in the same locus, they interact and they bring about the change in the phenotype. So such type of interaction is called as intralocus interaction, and since here the alleles are interacting, it is called as allelic interaction. Next is what? The interlocus interaction. So interlocus means what? The genes which is present in two different locus, they are interacting. Okay, the genes of two different locus, they interact and bring about the change in the phenotype. This is called as interlocus interaction. Since here the alleles are not playing the role, it is called as non-allelic interaction. Next we are going to study The types of interlocus interaction. So in types, you can say there are two types: epistatic or non-epistatic, additive or polygenic. So here epistatic or non-epistatic interaction means the gene which is present, two genes are there, two genes present in two different locus. One gene present in a locus, it suppress or hides the effect of another gene. Okay, so the gene which is dominating, that is called the epistatic. Okay, so here only the epistatic gene effect is being expressed there, character is being expressed there, so that is called as epistatic, and the character which is not exhibited, that is called as non-epistatic. Next is what? The additive or polygenic interaction. So here many genes together, they interact and bring about the change in the phenotype, then it is called as additive or polygenic interaction. Next we will study in the types of intralocus interaction. So they are broadly classified into three: dominant relationship. First one, complete dominance, as we studied in the monohybrid cross, we have studied example tall and dark tree plants, where it is crossed, the tall character is exhibited, that is complete dominance. Next one is incomplete dominance. Incomplete dominance means two alleles, they will show their expression there. Okay, both the alleles, they are not dominant, both the so then it is called as incomplete dominance. Both the alleles will interfere and they will give a result which is they will give a phenotype which is different from the parent, then it is called as incomplete dominance. Third type is codominance. Codominance means both the alleles will interact, both will show their presence. So both the characters are exhibited in the phenotype. And overdominance is the F1 progeny will be superior to their homozygous parents. Next is the lethal genes. Lethal means death. So here the gene interaction causing the death of the organism. So here when the dominant alleles are present, that is causing the death of an organism, then it is called as dominant lethals. Same way, when the recessive alleles are present together, they cause the death of an organism, then it is called as recessive lethals. Conditional lethals. Conditional lethals means based on the conditions like the temperature. So in one temperature, at low temperature, an organism can survive. In the higher temperature, the organisms cannot survive. So that is the conditional lethals. Next is sex-linked lethals. Next is the balanced lethal. Balanced lethal also, the heterozygous will be superior to the homozygous. The third type of interlocus interaction is multiple alleles. So one gene has many alleles, they are interacting and exhibiting the phenotype, then it is called as multiple alleles. So here Mendelian experiment proved that a single gene controls one character. This we very clear, very well known from the Mendelian work. We know that each character is controlled by a gene. But in post-Mendelian findings, various exceptions has been noticed in which different types of interactions are possible between the genes. This gene interaction concept was introduced and explained by W. Batson. So this concept is also known as factor hypothesis or Batson factor hypothesis. According to Batson factor hypothesis, the gene interactions can be classified as intragenic interaction or intergenic interactions. So as we have studied, intragenic means the genes which are present in the same locus, the alleles of the genes which are present in the same locus, they are interacting and they bring about the changes in the phenotype. Next is the intergenic gene interaction. Here the different genes present in different locus, they interact and they bring about the change in the phenotype. So first let's today study about the intragenic gene interaction. So intragenic takes place between the alleles of the same genes, that is the alleles at the same locus is called as intragenic or intralocus gene interaction. So it's clear here. So each gene has different alleles. The alleles at the same locus, they interact and they bring about the changes. So such type of interaction is called as intragenic or intra-locus gene interaction. So it has different types: incomplete dominance, co-dominance, multiple alleles, pleiotropic genes are some common examples of intragenic interaction. Let's study one by one each type with its example. So first one, incomplete dominance. Incomplete dominance here, no blending of genes. So this was first explained by German botanist Karl Korenz in 1905 in 4 o'clock plant, or it is the binomial name of this plant is Mirabilis jalapa. So here, when the pure breeding homozygous red, so it is represented as capital R, okay, so it is R1 is represented, okay. So here pure breeding homozygous is represented by R capital letters, since both are dominant, both the parents are dominant, we are using the capital letters. And next, so the red, homozygous red is represented as R1 is crossed with the homozygous white, okay. So here white, again a dominant parent, so it is represented as R2. So here both the parent plants, they are homozygous. The phenotype of the F1 hybrid is heterozygous pink. So here, when a Mirabilis jalapa plant, a red Mirabilis jalapa plant is crossed with the white Mirabilis jalapa plant, in the F1 generation, we are getting what? A pink color plant. Okay, so the genotype of the plant is R1. So the F1 heterozygous phenotype differs from both the parental homozygous phenotype. This cross did not exhibit the character of the dominant parent, but an intermediate color pink. So here, as we studied, when the red color parent plant is crossed with the white color parent plant, the offspring is pink color. So here it is what? Both the parental characters are not seen, okay. So but what is happening there? An intermediate color is obtained. When one allele is not completely dominant to another allele, it shows incomplete dominance. So here, neither we, if the F1 generation, neither we got the red flower, neither we got the white flower. So here it shows that none of the alleles is completely dominant over the another. So it is said to be incomplete dominance. Such allelic interaction is known as incomplete dominance. Okay. So here it's, I think so it's clear now. So here parent plant we selected a red flower and a white flower. When we cross in the F1 generation, we got the pink flower. Neither we got the red, neither we got the white. So this shows red and white, neither of them they are dominant. Okay, both are interfering with each other. So it is said to be incomplete dominance. So the F1 generation produced intermediate phenotype pink colored flower. When pink colored plants of F1 generation were interbreed, okay. Next, when this F1 generation plant, that is the pink flower plant, when it was self-crossed in the F2 generation, both phenotypic and genotypic ratios were found to be identical as 1:2:1. That is, in the F2 generation, we got one red plant, two pink plant and one white. Here you can see in this cross here. So if here the parent plant red represented by R1 and white represented by R2. So these are the parent plant. When it is crossed in the F1 generation, we got R1 that is a pink color flower, that is intermediate phenotype, pink heterozygote was obtained. When the selfing of this F1 plant was done in the F2 generation, we got one red, two pink and one white in the ratio of 1:2:1. So here the genotypic ratio you can see R1 is to R1 is to R2. This is the genotype. Okay, so three type of plants are obtained. Okay, so R1 represent red, the number of plants 1. R1 they are 2. Okay, and R2 is third type of white color flower plant. Okay, so this is the incomplete dominance. So here when phenotypic ratios were found to be identical as 1:2:1. So one red, two pink and one white. Genotypic ratio is 1 R1 R1, 2 R1 R2 and 1 R2 R2. From this we conclude that the alleles themselves remain discrete. So this shows that the alleles, they never blend together, they never mix together, they remain unique, they remain pure, discrete and unaltered, proving the Mendelian's law of segregation. As we have studied law of segregation, we have studied in that the blending of the genes will not take place. They will be remain as a hidden form, but they may reappear in the next generation. So that is the law of segregation or law of purity of gametes. The phenotypic and genotypic ratio are the same. Okay, there is no blending of genes. The genes are not the alleles of the genes, they are not mixing. In the F2 generation, R1 and R2 genes segregate and recombine to produce red, pink and white in the ratio of 1:2:1. Okay, now here we are going to study the reason why when a red color flower was crossed with the white color flower, we got the pink color flower. So here the R1 allele codes for an enzyme responsible for the formation of a red pigment. So the R1 allele has an ability to produce a functional enzyme which promotes the formation of a red color pigment, whereas R2 allele codes for defective enzyme, whereas the R2 allele is not synthesizing this functional enzyme. So when R1 and R2 genotypes produce only enough red pigments, so when R1 is present, it will produce enough red pigment, but only when in the genotype R1 R2, this R1 will produce only 50% of the red pigment to make the flower pink. So two R1 are needed for producing red flowers. Two R2 genes are needed for producing white flowers. If blending had taken place, the original pure traits would not have appeared and all F2 plants would have been pink flowers. So here it is said that if both the R1 is present, both the alleles R1 is present, we'll get a red flower. If both R2 is present, we'll get a white flower. But since R1 only one R1 is present there, that R1 synthesizes only 50% of the red pigment. As a result, the color of the flower is pink. So here when R1 R2, the alleles are not mixing. As a result only in the F2 generation,